I'd like to get started today. This is the July 8th meeting of GLIMPRINT. We have a seminar today and our usual introductory material. As always, I need to remind people that this meeting is live streamed and recorded and available in edited form publicly on YouTube after the fact. As always, Tom Helikar, myself, Lorenzo, and Ellen are available to take your suggestions, critiques, comments, anything that you think we can do better. Please let us know. Our email addresses are here. We're happy to hear from you. If you find the seminars interesting and you'd like to hear more on related topics, please consider joining GLIMPRINT. I'm going to have another slide on that. And subscribing to our YouTube playlists. And there are links available in the information at the bottom of this video when it's recorded. And we can also provide those here during the meeting. GLIMPRINT is a small membership organization. We would love to have people join. Individual memberships are $100 a year for faculty and staff, $10 for students. If you want to become a member and you need a fee waiver, we can do that. We would love to have some institutional and corporate memberships. So if any companies would like to help us build this community, we would love to have your support. And again, please contact us if you're interested in doing that. We have a variety of social media links. There's the IMAG MSM wiki page. There is the GLIMPRINT page, which thanks to Jacob Barak is really nice now. We have the YouTube GLIMPRINT seminar archive, which has more than 170 talks from the series. There's a LinkedIn website and less used are Slack channel and X channel as well. So please help us get the word out and build this community. If you have a publication to share and you would like to have that put up, we can do that on the IMAG site and also on the GLIMPRINT site. On the GLIMPRINT site, there's actually a nice user interface for submitting your information. Send that to ellenmq@iu.edu if you need help with the GLIMPRINT, the IMAG site. And if you want help with the GLIMPRINT site, Ellen is also your point person. We can announce conferences, we can announce job openings, grant opportunities, recent publications, recent software products, anything you think the community would like to see. Please help us put that up on the GLIMPRINT site. Coming up next week, we have another one in the Lessons Learned series that Jacob has been pioneering from lessons from COVID-19, data trust and decision-making in times of crisis. And then July 23rd, we have Andrew Sage, Digital Twins of Ex Vivo Human Lungs. Then we will be taking the month of August off. And starting in September, we will go back to bi-weekly meetings. We will be changing the day of the week of the meeting by popular request from Thursday to Wednesday. It's also for the benefit of our West Coast members. We're moving the time a little bit later. It's going to be starting at noon Eastern time on Wednesdays. So a day earlier and 2 hours later. And we have 2 talks already on the books. September 9th from Queensland, targeting oligodendrocytes in an Asian-based model of multiple sclerosis. And then Jupiter Algorta, September 23rd, is going to be talking about a data-driven model of polarity reversal in migrating cells. Jupiter was the winner of the OpenVT reproducible modeling competition this year, and he's presenting that as part of the recognition for his work in making models shareable and reproducible. If you would like to suggest speakers, you can do that on the GLIMPRINT site. You can also directly email Lorenzo Vischini at gmail.com. who does coordination for this. He's very effective at getting people to say yes if you invite them. You could suggest yourself, you could suggest your postdocs or your graduate students. Don't be shy if you've spoken before and you think that you would like to speak again on the same topic or new topics. Please also feel free to suggest people you'd like to hear. Those people could be ones that have worked in areas that you find interesting, a paper that you've seen that's interesting to you. They could be in basic research, they could be in computation. It doesn't have to be immune digital twins strictly. It could be drug development, it could be biotechnology, measurement, computing, anything that you think is relevant to the eventual goal of immune digital twins and immune modeling. Okay, today we have a talk beyond endpoints, why imaging is most direct window into cell biology. Topic, I think, of great interest to many of us, and we're looking forward to hearing about that. And I will turn the screen share over to you.
Hello, everyone. It's a pleasure to be here amongst fellow researchers. Uh, I know most of you guys are probably or mostly familiar with microscopes, but what I want to do tell you is just like everything else in our lives, microscopes are also getting automated. Technology is very ubiquitous. It's touching everything. In our lives, and microscopes are not an exception to that rule. Um, Hello again, everyone. My name is Sesan Ajina. I am the co-founder and CEO of iLABS, and my talk today would be about why imaging is the most direct window into cell biology and how that compares to endpoint assays and what the future potentially looks like right now. Um, Everything in nature is in motion, and biology is not an exception to that rule. Biology is dynamic. It's not a snapshot, it's a movie, and every biological process is constantly in motion. Um, how we measure that is very important because as a researcher, that gives you a window into what is it exactly that you are trying to capture and what stage in the biological process actually makes a lot of has a lot of direct impact to your research and your understanding of what that context of the biological process is actually about. Cells are constantly dividing, migrating, or differentiating. Um, and the way that you capture that really drives your understanding of that biological process. Um, Endpoint assays are really nice. They're good. They help with a lot of validation. They help you better understand what's going on with your cells. And if we think about like the roles that cells play in overall biology, it's very critical. As the foundational unit of life, understanding what goes on in your cells is a very critical issue when it comes to solving diseases, which is our focus. And the way to better do this is to have very detailed and foundational understanding of these behaviors. Um, the limitations with endpoint is that they give you one readout, one measurement, one number. Uh, and it always happens at the end when it's actually done. So that way you can answer critical questions like, um, did the cells stop dividing? Did they die? When did they die? Did they migrate? If they did, how often and how long did they migrate for? what's the mechanism that actually underlines this behavior? These are questions that endpoint assays cannot answer. Um, I totally understand that most researchers are used to this perspective, but what I usually try to convey is that the way you see a picture is different from how you would view a movie. One gives you a lot more information, and the other just describes a current situation. Um, Different biological mechanisms can produce identical readouts, even if the underlying biological context is different. And I would say more about that in subsequent slides. Um, Imaging is a very, it's It's the preferred method for some key reasons. First, it's non-invasive, it's non-destructive, meaning you retain your samples, and you could get multiple readouts just from one single samples. Like I said before, because microscopes are getting automated, you could collect images of cells much more quicker than you would before. So an example is like 5 years ago, let's say your research lab or a research lab that's generating 5 slides a day. Now that same lab can generate 500 slides a day because you have an automated image acquisition and experimental protocol that makes all of that much, much quicker. So you get images from your cells in non-destructive and non-invasive way. You let your cells exist in their natural habitat, and that way you could observe and quantify their behavior on a single cell level or in population statistics if you're trying to group clusters depending on what your research approach is. And from that, using uh, uh, pre-trained and fine-tuned AI models, you could interpret biological information just from your data set. And if this is done properly, each of your experiments would be fine-tuned for an AI model. And what that means is that for every experiment that you run, you would sort of have like an AI model that's dedicated to that, so you can guarantee reproducible analysis, because it's not science if it's not reproducible. And what imaging helps you do in this context is it gives you like a direct window, direct rich window into cell biology as a as opposed to um, endpoint assays, which is a process of inference. You know, you are trying to correlate some biological readings to a very specific interpretation or readout. Uh, a really cool thing that imaging gives you as a benefit is uh, all the way from seeding your wells to the end of the experiment, you have all of that information. So you are uh, labeling of your cells, labeling in terms of like, you could think about it as giving all of the cells in your experiment an ID, so like an ID tag. And you know each one by their label, by their name, their ID index, and you can track each of them throughout the course of your experiment. And with endpoint assays, you get the results at the end of the experiment. In this example, a 24-hour growth assay, you can see what happened at the end of the experiment. However, imaging gives you like a continuous observation of each cell and what the behavior is. And we are asking not just what happened, but exactly how it happened. Uh, with this information, this rich information that you have, you could quantify multiple assays. Uh, with endpoint assays, one assay is one reading and it's for one particular uh, uh, analysis. With imaging, you could have multiple readings, multiple readouts just from one data set. You could have morphology, intensity, cell cycle, even subcellular structures. And the key thing here to make this really work is making sure that you standardize your imaging protocol and your experimental protocol, and you make sure that you validate that process. And as long as you do that diligently, you can have one data set that would provide lots of biological readouts down to a single cell resolution level. So this makes your biological measurement a lot easier, and you can quantify incredible details just from how your cells are behaving throughout the course of the experiments. Uh, Here is what I was talking about before in terms of like having the same readout and different uh, underlying biology. The biological context is totally different. So this is an example of testing two different drugs on a um, sample. And even though the sample has the same readout from an endpoint perspective, uh, the underlying biological context is very different. The morphology is different, and morphology says a lot about how your cells are responding to your therapy or your drug or your perturbation. And I will say more about that in terms of phenomics. And migration, it says a lot about the energy in your cells. If you have higher migration rate and the distance covered, that means there's a lot of energy with your cells. The cell cycle phase, how you measure mostly subcellular structures. The power of their intensity says a lot about the cycle and the chromatin and the DNA and what's going on, especially during uh, uh, division cycles. You could actually measure how long it takes for the cells to divide, which signals a very important information about the cell cycle phase and key underlying behaviors for your cells. And the phenotype, you can measure how the phenotype affects the behavior and the clustering of all of your cells. Uh, AI is a critical tool, and I'm sure a lot of uh, folks here I've heard about how AI would affect science. I have some opinions about that, but that's just for another day. But AI is a very important tool. The key thing here is knowing how to use it and apply it in the correct way. Um, like I said before, microscopes are getting automated. So you have image stacks now instead of single slides. Humans can't analyze image stacks. That would take forever. So if you can build an AI model that's specifically designed for your experiments, a model that understands your experiment, you could automate a lot of these workflows. You could do segmentation, which is, you're separating your cells and indexing them and giving them that tag, that label that I spoke about. You could track them. So after you actually label them, you could follow them throughout the course of your experiment. You could extract features as you're tracking each of them, and you could observe the cells in their um, natural environment and just observe patterns. And from these patterns, from this clustering, you can quantify key behavior. Um, the real uh, goal here is that you can analyze lots and lots and thousands of cells that as a human being you would typically miss. So AI is definitely a powerful tool that helps to get all of those kind of tasks done. Um, and Phenomics. I'm not sure how familiar folks here are with phenomics, but it's something that's been around theoretically as a concept in cell culturing for a while, but it's now getting to a stage where it's becoming real and been applicable. Uh, Omics is awesome. Multi-omics. We have all of these different types of omics that drive how lots of research have been done today. But where genomics is different is I, I like to think of it as reverse engineering for biology. So instead of trying to uh, design a gene or an RNA that would change something on a foundational biological level, you are observing cells in their natural environment. You are observing what they do. And because you have this powerful AI that can interpret different kinds of patterns, and you can quantify cells in a healthy state and a diseased state, uh, Phenomics would help in testing lots of compounds, lots of drugs. Uh, because you're not sure exactly what drug or what compound is going to work, and a lot of folks actually start with like some in silico perspective, where you predict how some compound is going to change uh, uh, foundational biological process in cells, you have the ability to run thousands of experiments, to generate thousands of slides and images. And with phenomics, you have a model where you've quantified a diseased state and a healthy state, and you watch which compound actually moves the cell to a healthy state. And if you can quantify that behavior, then you can use that as um, uh, uh, a hit generation. So you could use this to find drugs that would actually move the needle. And once you have that, you could quickly build this in vitro models that would help you accelerate the path to clinical trials. Um, with phenomics, you are quantifying cell behavior, and you have a model that actually helps you interpret what that behavior means. And once you get a behavioral um, output that matches a healthy state, then you can uh, proceed to move the compound down the funnel as one that's promising. Uh, it's very, very novel, and I will be happy to talk to researchers more about some of the tools that we're building to solve this. And it's something that's very, very interesting and probably the future of solar analysis. Um, a critical benefit of this application and of imaging in general is that it could be applied at different stages of the pipeline. Um, from target discovery to screening to understanding mechanisms of action, you can actually apply this in different contexts. So imaging is a critical tool if you're not already using it, or you're not using it in a much more high throughput context. I would really uh, uh, I would really advise that you consider, because the barrier to entry for this is super low now, and a lot of the tools that you need, some of them are even free, and there is no reason for you not to want to try this out and see what it would actually do in your experimental workflows. And that brings me to iLABS. Um, uh, we are a um, company that's trying to build the foundational infrastructure to help researchers process all of this massive image and data sets. Our goal is to make access to all of this powerful AI tools that I was talking about accessible to everyone. And the way we are doing that is by um, making this a simple web-based tool. So instead of like in the past, where if you want to use a powerful AI imaging tool in biology, you have to know like lots of programming and code. No, as long as you can use simple tools like LinkedIn or Facebook, you could use the tool that we're building. And the way it works is very simple. You generate your data, and once you have your data, you upload your data set. And once you upload your data set, we would apply a fine-tune model. We would fine-tune a model that's specifically for your experiment to make sure that it's reproducible. And once we do that, you could now automatically run those key um, analytical workflows from like segmentation to tracking all of your cells. And from there, you could read multiple rich outputs like growth, morphology, motility. And a lot of our in vitro workflows are designed specifically for your experiments, including like the critical readouts that would make interpretation very straightforward and easily understandable. We have interactive dashboards that makes visualization very simple, and we are currently in beta. So if you are interested, please reach out to me. I would have my email at the end of the presentation, and we have a sample data set that you could initially play around with just to get a quick understanding of how this works. It's cloud-based, it's GPU accelerated, and there is no coding experience required. Um, the reason that this matters is that there is a transformation going on right now in biology. Uh, I'm not sure if folks are familiar with FDA's Modernization Act. There is a push from the government, from FDA and NSH, to have new in vitro systems. Uh, there, this push is aimed at moving away from animal models because animal models don't translate properly in clinical trials. So having human relevant in vitro models is now getting a very big push from the government. What we are trying to build now is the foundational infrastructure that would help that transition to the new in vitro system. And that way, researchers can get more from their experiments. They can have better levels of sensitivity. You can quantify cells on a single cell level, detect very, very subtle changes. You reveal hidden patterns from a phenomics analysis, and you can actually see how diverse or not your cellular population is in terms of their behavior. You remove bias from your analysis because you've handed your analysis over to a fine-tuned and well-trained model that all it does is just measure your cells and looks at your experiments to understand what's going on. And this drives a lot more faster decisions. So you can make real quick decisions about how your cells are behaving, and these decisions are based on um, big sets of data that accelerate how you actually make new discoveries across different domains. So during my preparation, um, I believe it was Lorenzo who reached out to me to talk a little bit about uh, how to start a biotech startup. And I wanted to say something about that. Um, a caveat here is that, we are in the process as well. We're learning a lot. But I would like to share some of the things I've learned so far and see how, or hopefully see how this could be of help to folks. The first thing is that the journey is really linear. Actually, it's never linear. It's a very squiggly type of journey. And the way you think it's going to happen is actually not how it's going to happen. Mostly founders think, you build the technology, you get some funding, then you get customers, and then you scale real quick, and everything is awesome. But that's not how it happens. At least from my perspectives and from the things that I see, the first thing I would say is that you have to find a very um, niche problem. Niche in the sense that it has to be very narrow. And the reason it has to be very narrow is uh, as a startup, you have limited resources. And if you do too many things, you're going to spread yourself thin. So you focus on a very niche problem. Uh, that's the first thing. Secondly, is be ready to um, pivot repeatedly. Um, Pivoting is kind of a dance. And the more you interact with your users, the people that you are serving, the better that is. And it's that process of interaction where you get to better understand their needs and you try to calibrate and build to match those needs. And in bio, of course, you have to be mindful of regulations. I just spoke a few minutes ago about FDA and NIH. You have to be mindful of what's going on. In our case, at this stage, we are research use only. But if you're building for clinical applications, you have to be mindful of like regulations and how that pertains to your startup. Uh, the the other thing is talent. You have to work with people who are genuinely interested in what you guys are working on. You have to be a collective group of talented people who have a mission of what you want to accomplish, and that has to be reiterated quite often to make sure everyone is on the right page. And of course, you have to manage cash because, even scientific research works on money and build credibility. Credibility is a critical currency in biotech. And if you are not credible or not forthcoming and truthful in your approach, that would be a bad omen. Uh, so how to prepare, I would break it down into the simple steps is talk to your customers regularly, like all the time to make sure that you have a pulse of where they are. Focus on critical pain points, build a strong scientific network, um, protect your IP. This could be critical uh, down the road, and plan for like a very long runway, preferably like 18 to 24 months of runway to give you a lot of space to meets all of these critical milestones and deliverables. So the biggest thing here is that um, uh, it's the science is usually not the issue. It's building something that solves like a real urgent problem for specific and targeted researchers. And I hope that this helps. And I'm looking forward to all of the wonderful projects and startups that are going to come out from folks here. And yeah, if you have any other questions regarding this, of course, I'll be very, very happy to answer them. So thank you very much for your time. I really appreciate you guys inviting me here today. I hope that you would consider having imaging play a bigger role in your experiments overall. And looking forward to all of your questions. And yeah, thank you.
Okay, thank you very much. There are some questions from the audience. Ellen, go ahead.
Yes. So I've done a lot of imaging over the years and used several different microscope systems. And I'm wondering, I might have missed this. Are you working with existing microscopy companies and the imaging equipment companies and building systems that then go from the microscope all the way through the imaging process as well as the image analysis? Or are you just starting with images that are coming off of these systems and then working through the image analysis aspect of things?
Yeah, thank you, Ellen. And yes, so we are a post-acquisition analysis tools.
Okay.
So typically, the researchers that we work with, you have like your imaging facility, your core facility. You go there to generate your data, and once you have your data, you you would upload that to our platform for post-acquisition analysis. But prior to that, we would have worked with them on like some validation steps just to better understand your experimental protocols so we can fine tune that model, right? Because that model has to be fine tuned for your experiment. We don't do like a cookie cutter approach. Like it's very, very tailored to experiment to make sure that the readout is reproducible. I hope that answered your question.
So another question I have regarding this is since companies like Nikon, Olympus, Leica Microsystems, all of them have their own image analysis and acquisition packages that are pretty sophisticated, frankly, and go all the way from time-lapse imaging, static imaging or time-lapse imaging, certain ways that they stack things and measure things and tools for all of that. How are you working with all of those companies to make sure that when you are taking their image format, because of course the proprietary imaging format that
Right.
you're saving in is where all of the data is. And if you start saving that proprietary imaging format into some other imaging file type, you can lose or mess up some of the really important information that's saved in that file format. So are you working with all those microscopy systems, and are you making sure that you're able to translate from their system into your platform without losing information?
Yes, yes. Another wonderful question. So the first one is yes, we work with lots of microscopes, so we are microscope agnostic. We've actually worked with Nikon, we've worked with Thermo Fisher, Edoluma. molecular devices. So we work with like lots of microscopes. And in terms of format, you are very right about that. Some microscopes have like, their own specific formats. But what we do is we convert that. So we can work with like TIFF, OMER, and PNG. And the conversion is a it retains all of the information of the data. And the reason why this is important is that it's that information that you're going to quantify later downstream. So you don't want to lose anything. It's very important. We personally, we prefer TIFFs because TIFF is just like the raw tagged format where everything is intact. And in terms of like the microscopes also having their own software, yes, that is true. Actually, all of them have like some
Yeah.
form of analysis. But the problem, though, is that this software for the instruments are designed primarily for acquisition. They do some analysis.
That's not actually true. I'm sorry. Nikon and Olympus and Leica Microsystems actually have quite sophisticated software. You really
Yeah,
should make sure that you discuss with all of them. And if you're able to, I don't know if your company buys systems so that they can become more familiar with the post-processing aspects and the actual image programs that exist out there. But I think if you haven't recently, and I'm thinking within the last 10 years at least, you should definitely talk to these other companies about their imaging software or investigate them because they're full systems now, fully integrated systems, and they're really pretty amazing
yeah.
what they can do.
I do agree. Yeah, it's very, a lot of the systems have like a very interesting analysis. But what I mean that primarily is the first the first thing that they do is the software has to be able to take data from the instrument. That's like the core thing that the software does. Because if you can't do that correctly, you can't even analyze. So a lot of their engineering effort, because I've worked with lots of this folks, a lot of their engineering effort is getting that data. And for analysis, yes, they do. They have like all of this analytical tool. But what we offer to them is number one, reproducibility. And the challenge there is that because they are focused on building that software to work very well with their instruments, which they do a lot well, they can't guarantee reproducibility. So if you use that analytical software, the one you were just talking about, for example, and you share that data set with someone else that uses their analysis, you're not going to get the same results. And this is like something that has been tried a lot. And the reason for that is I was talking about like building models for your experiment. So the way you do segmentation, for example, Nikon specifically, the way their analytical tool works is they use something called thresholding. So thresholding is like you're doing like pixel contrast level and you just cut them off. So if you had like 100 cells on your screen, legit, maybe you had like a legit 100 cells that you want to identify, it would get maybe 54. So you're still missing out on a lot. And that means that you cannot reproduce that data. Even though like the workflow and everything is super sophisticated, you are missing step zero, meaning you can accurately tell me the number of cells that you have in your experiment. So that's like one of the core things that we offer to folks like Nikon with their sophisticated analysis is the models that we build. So our models are built in-house and it's specifically designed to quantify human cells. So that's like where we have this differentiator, where we have this model that's benchmarks as the best in identifying and quantifying human cells. That's the only thing that we do. We build a model that's just looking at human cells to better understand what it does. And regarding like the interoperability, another thing we do is like offering this on a web platform, so you don't have to download anything on your computer. You don't have to install anything. All you have to do is just log in. If we've worked with you before and we've fine-tuned a model for your experiment, for example, all you would have to do is log in, grab your model, take your data, and just start getting your results. Yeah. I don't know if that answered your question.
Well, partially, but I also have a rebuttal if you care.
Oh, okay, please,
Um,
please, please.
so thresholding is one of many different ways you can segment cells. I mean, and that is
Right,
not typically, that's like the most rudimentary method, to be honest. And so
right,
that's not typically, I mean, that's the first method most people learn at the very beginning of time, but it's not typically the method they use when they're more sophisticated at image analysis. One suggestion I would say for your company is if you could actually integrate with a company like Nikon and their software and get that whole workflow paired up with a microscopy company, I think you'd probably make a lot of money, to be honest. Maybe more money
yeah.
than you may make being on your own, to be honest.
Yeah, actually, Elliot, we totally need to connect after this. I would like to, yeah, talk to you a lot more. But
Mhm.
yes, we are, the problem with like big corporations like that is number one, they move slow. And number two, is that, integrating into their workflow would could be challenging for them, not for us, because it will mean that they would have to move stuff around, and sometimes you
I
don't
bet
even.
you not so much, to be honest.
Oh, okay,
I think
okay,
you could integrate with all these companies a lot more effectively than you realize. So,
yeah,
wow.
we definitely need to talk more after this. But thank you, really, really appreciate the question. Yeah.
You say that your value is the model and the reproducibility that's out there later on, and you are using the new AI models, if I understand correctly, from because you're saying you're using GPUs and stuff like that. There's one problem: those AI models are not reproducible. Not that potentially you can make them reproducible to some degree, but the fact that once you're using a GPU, then this reproducibility is only theoretical. Because once you run things in parallel, you never know how to, when they come up back, how you add them up. And on GPUs, they kind of run in random numbers. There's no way to force the GPU to run them in a sequence that will be the same. And therefore, the results that come out later on are marginally different each time you run them. So now my question is, how many times did you actually encounter this small arithmetic problem of that caused by polarization actually affects your models, if you ever seen it? And B, do you have any ways to mitigate it?
Yeah, thank you very much, Jacob. We've seen some drift sometimes, and we've actually built a layer in our model that addresses the drift in performance sometimes. But the way we fix that with like a human in the loop approach is to have validation checks every now and then. So you do like a validation check for maybe every 3 months, just to make sure that the model is performing at the optimal level. And this
I,
validation check,
I'm not talking about model drift. I'm more talking about the fact that once you train a model with
right?
today's hardware and so on, you cannot guarantee reproducibility to the very exact numbers. There's always some sort of small little error. You just cannot. It's a small numerical error, but it can become bigger. So my question is, did you notice it? Whenever
Uh,
you claim reproducibility, did you run tests to actually see that it exists or not?
so the way we validate is by using like benchmarks. So like, IOUs, for example, and if we pass like a certain benchmark, say, for example, 0.9. That's usually acceptable. So if the error is still below that threshold is typically good. It's when it drops that we'll be like, okay, then there's an issue here. So maybe another round of fine tuning is necessary.
Okay, you're addressing it statistically. Your answer to me means you haven't encountered this phenomena, but maybe it's not.
Maybe I'm not understanding your question because
it's a very minute issue. The fact of how it's okay. It's like, but if you haven't noticed it, like if you run, you claim reproducibility. So when you actually run the same model twice, you'll never get the same results to a very slight number difference. Check it out.
Yes.
Check it out. Now,
yeah, but if
Jacob,
that difference is below the threshold, right, it's still considered good because the threshold is like is the pass go point.
You're reproducible up to a statistical error. Got it. Thank you.
Yeah, thank you.
I think the issue here is Jacob is asking a technical question on numerical reproducibility, but in fact, in microscopy, the issues of reproducibility and noise are so large that it seems that it's not impossible, but it's probably unlikely that the specific type of reproducibility issue Jacob is concerned about is going to be the most significant one. I was more interested in another aspect of reproducibility, which is you talk about automation, but when you're doing post-processing, you're not actually automating the workflow for microscopy, if I understand right. The person will, or the lab will do the time series analysis using whatever protocols they use, and then you are going to pass the time series data set, which may be two-dimensional fluorescence or scanning confocal, it could be two-photon, or it could be SPIM, I suppose, back to your online platform for analysis. Is that right?
Yes, that's correct. Post-acquisition in different timelines.
So one of the things that you run into it's you're already doing a lot, so it may not be the thing that you care about the most here. Solving any of these problems, even one of the many problems you list, is already a big deal. Is that if you are going to automate microscopy, you have issues with, and thinking of Ellen, I'm going to say that the current generations of microscopes have gotten better at this. You have issues with loss of focus on the sample because of sample drift. You have issues of bleaching of the fluorescent dyes so that intensities change as you make successive measurements. If you're going to do truly roboticized microscopy, where you have to change samples repeatedly automatically, you have issues of registration for the same sample, so that you could say that the cell number such and such in image such and such is the same as cell number such and such in the following image. And again, those may all be outside of the remit of the particular approach you're taking, which is post-processing. But I wondered if you're thinking about those kinds of reproducibility issues, which are really microscope-facing reproducibility issues, as well as the post-processing reproducibility issues.
So a lot of like these modern microscopes that we work with, they have this robotic thing nailed down. They have like, you even have experiments where you are like 3D experiments where you're quantifying at different focal planes, and they do that really well. These robots have been programmed and designed that they have like their focus nailed down. And for like the Nikon, for example, who worked in Nikon, it's really good at that. What we do is when it comes to like the post-acquisition is we have a pre-processing stage where we analyze all of these variables to make sure they are good. Because if they're not, then you have like the wrong inputs. So what we try to do in some of those with some of those pre-processing tasks is correct for some of these variations, using different types of logic, but sometimes we can't, it's just too bad and we can't correct for them. And then you would need to have like a better data input. But the modern microscopes, they don't actually have these problems anymore. They used to, but if you get like a really good one, and I could make recommendations because we've worked with lots of microscope, different types, even ones that like you have weird problems with some where you have like humidity, where the thing is fogging up, so you can have like different types of technical issue. But yes, some of these modern microscopes, they're very good now. They're really good.
And so you're basically hoping that your software can handle things like intensity calibration and registration correction, post-processing, whatever the microscope hasn't done automatically, you're going to take care of. That
Y-
makes sense. That's fine. Nobody in this audience will ever object to your emphasis on dynamic measurement rather than endpoint measurement. In fact, almost every talk I give, uh, and I give hundreds of them, starts out by making a statement that you can't understand biological dynamics with endpoint measurements. You have to do longitudinal dynamic measurements. And so your philosophy is one that I find extremely appealing. And the things that you're trying to do are things that I think the vast majority of people in our community would identify as critical. The one thing that might be a bit of an issue, and I understand the appeal of going to a online post-processing system, getting the post-processing burden off of the host computers, getting it out of the microscope. But the one thing that you do lose is some of the flexibility and power of doing imaging in real time, which is that if you have real-time analysis of what you're seeing on your microscope platform, you can have experimental protocols that depend on what happened. So, for example, if you wanted to have an active control strategy that involved imaging, then the analysis of the images would have to be done in real time in registration with the experiment. This is a community that thinks a lot about digital twins, and so we're very concerned with building real-time replicates of specific instances of experimental systems so that those computer simulations of the experimental system can run in real time in register with the experiment. If you're going to have those digital twins, you need to be able to rebase them periodically during the experiment and in response to the experimental intervention. And that means you need to be able to do the analysis on the fly too. I suspect you could build a pipeline where instead of collecting the entire data set and then passing it off, you could pipe the images as they become available to your workflow. But I wondered if that's something that you're working on or thinking about, because I think it would greatly extend the power of some of the techniques that you've described to do the specific kinds of interventional, for example, the toxicological or interventional studies that you're talking about.
Yeah, yeah. Thank you for that. We have definitely played around with that before, making it real time. But we ran into some technical problems. And the problem was communication protocols, all of this microscopes, they, like Ellen was saying earlier, some of them have their own formats and different stuff going on. Part of their customization is like a communication protocol, and integrating to that would be challenging if you can't properly get data from that particular protocol. It's something that we're thinking about trying again, down the road, but we are just focused on where we can add immediate value right now. And the immediate value is when researchers, the way that it's currently done is researchers take their data once it's generated to some try some open source tool or try some like post acquisition analysis script, and we want to make that process easier. And that's a place where we believe we can add value right now, which is why we are focusing on that. But this is still something we are planning to try again later down the road, hopefully when we can easily have all of these conversations with these microscope companies.
Well, I agree with you. And data flows can be dramatic, maybe not so much in classic fluorescence imaging, but when you get to, and two photon can be fairly slow too. But for example, SPIM, the limitation of the best SPIM systems is actually the pipeline from the imaging to the hard drive where you're going to store the images, because the SPIM actually generates data faster than you can store it onto a hard drive. And so I understand the issue that it takes time to do these computations, and therefore it may not be practical to do them in real time yet in all cases, and that starting with a deployment like the one that you described is a reasonable first step to bootstrap.
First of all, what's a really simple question? What's the I in iLABS stand for?
Thank you for that. Imaging lab.
Imaging, thank you.
Yes.
Yes, a one-word answer. Okay, and I always have this vision of a biology lab having only three things figuratively. The first is a microscope. The second is a petri dish, and then a computer now. Okay, and what you're showing us is the capability to extract a huge amount of information from the microscope, which is the analog real world imagery, into a form that the computer can use somehow. And so that has limited us for a very long time. But I've always been equally interested in, I have a model that the data from the imagery from the analog experiment is given to the model, and the model doesn't quite work under those conditions. What it's seeing isn't quite right. The model says, the model can't make sense of that, so I'm not right yet as the model. But at the same time, the model may say, there's something missing in the information that I have. Okay, that my model almost works, but something is missing. And in order for my model to be right, there would have to be something happening in the analog world like this. And now we get to the human with the microscope who says, well, if that existed, I would have to treat the tissue sample differently to expose something like that. But since you, the model, have suggested it might be there, I'm going to try to come up with something that might enable the microscope to see exactly what we're missing. And so what we're talking about is it's a two-way kind of street. Are we making progress on the part where the model suggests to the microscope people something to be looked for, which the human never thought about, but now that I know what I'm looking for, I know how to set up my microscope, change the settings, change the processing, and oh my God, look at what we just found. How are we doing on the potential for the two-way between the microscope analog world and the digital? And it's you're making a lot of progress on the analog to the digital here. How are we going to do the other way?
I will try to reiterate your question to make sure I understand it. So what you're saying is that how is the model, or can there be a model that would understand your experiments and suggest some new approach, or maybe give you new insights into what you should do in terms of like a uh, research perspective? Am I saying that correctly?
There's something is happening that we just don't understand why yet, and it's because we're not looking at it analog, looking at it the right way. We're not doing anything that would expose that chemical transmission between two cells. But if I know what I'm looking for, I can treat it, and then you say, "Oh my goodness, look at that! They're talking to each other."
Mm.
And now maybe the integration of the model helped me find it. We now understand it. Simpler would be any of the things that the model predicts that you should be able to see. If we look and it's not there, is the model right? Well, maybe we don't know how to look for it, but maybe no, the model is wrong. It doesn't exist. That what your model assumes or what your model would require in order for it to work is something that isn't there.
I think what you're talking about is like a reasoning model, something like, an LLM, or like the new Fable, something that actually interacts with your data and analyzes everything, and you're kind of like talking to it, and it's connecting different aspects of the experiments to other parts of previously existing knowledge.
Right.
That's a different model. That's a reasoning model. And that's definitely a future direction. What we are building right now is a very specialized and niche model that's like trained for just one task, quantifying the behavioral um, patterns of your experiment. It's like, you train a dog to do one thing. That's the kind of model that we have here. It's trained to just look at your cells in your experiments, label them, track and quantify their behavior. In terms of like the sophisticated reasoning model, that sounds like a very interesting project. It's going to take a lot of work and a lot of clear definitions of what the outcome should be, but it's definitely an interesting project. It's an interesting suggestion. I'm not really clear though how that would be connected to like other layers on a reasoning level, and how those layers will be built on existing knowledge. But that's like
Yeah.
something like core scientists would have to specify.
It's something I think, it's future, and there may be people with other questions now, but I'm gonna want to know where you are and how to
Oh,
get home to you. Okay.
I am totally for it. I'm
Yep.
totally for it. And sounds very exciting, sounds very futuristic. And if you can have a model that can look at your experiments and tell you all of the things you think you're missing in there, the things you
Yes.
should add. And yeah, that's
Yep.
definitely something that I want to see in the world. I definitely want to see that in the world.
Okay, thank you.
Right. Well, I think what John is discussing and what I was talking about in terms of the real-time feedback are, as I say, I understand why that's something that may be a step too far to begin with. It's certainly something that there are a bunch of ARPA-H calls for projects that involve that kind of coupling between imaging and analysis. And so maybe in the next few years, when computing gets faster and AI gets more reliable, then we'll be able to do those things. You sounded like you were proposing, though, more of a CRO model for your company, where you're going to develop a bespoke analysis pipeline for each client. Is that correct?
Yeah, we develop customized pipelines, but I wouldn't say a CRO because a CRO would entail that we're doing like the analysis for you. We're not, we're just interpreting what's going on in your experiment and making sure that's reproducible. The science and the biology, you would still have to make those decisions.
But thank you very much for the clarification. Okay, well, I want to thank you then for coming to speak to us today, and good luck on developing the company forward. I think you're trying to do things that we all believe in very much, so we would love to see new approaches to delivering this kind of product. Thank you so much, and we'll see people in 1 week, not in 2 weeks this time.